Thank you everybody for joining the call. And I would like to talk about today our ZenoTOF 8600 system for especially can be used for single cell proteomics. It can be used for different other application, but this talk is around single cell proteomics analysis. So SciEx HRMS platform, there are mainly X500 series platform and also ZenoTOF 7600 plus system and also ZenoTOF 8600. These are the HRMS families SciEx having. X500 system, we have X500R and X500B. R system is mostly used for small molecule or like peptide and routine peptide analysis, and this is very easy to use system. And X500B system is used for biopharmaceutical characterization analysis and routine like small molecule lipids metabolites, all analysis can be done by X500 system. So this is a tabletop system, very small footprint, easy to use, and it has a lot of different good software solution like SciEx OS for data acquisition and data processing. And this also having a twin spray turbo V source that can be used for data acquisition as well as TOF calibration, automatic TOF calibration. It also includes SWOT DIA acquisition option in the software. Then we have ZenoTOF 7600 and 7600 plus system. 7600 plus system having ZenoSWAT DIA acquisition, EA defragmentation, ZenoTrap technology, and JetTscan DIA 3.0 acquisition. So these are the additional option you will get in ZenoTOF 7600 system. In addition to that, we have OptiFlow source that is very much useful for switching from nanoflow to microflow. That means any application like proteomics application, especially use nanoflow, there we can use the OptiFlow source. And if you want to use this system for metabolomics or lipidomics application, same OptiFlow source can be used. So this is giving better like option or flexibility to the user. You need not to break the vacuum. So that is one of the best part from proteomics to the metabolomics analysis. So it reduce your runtime or your high throughput analysis is possible in both the application. 8600 is this system is launched in 2025 in ASMS, and in 2026 8600 EVO also launched. And this system is a very high-end instrument. It offers very high level of sensitivity and also in both MS and MSMS level. Apart from this, ZenoTrapping EA defragmentation, ZenoSWAT DIA, and JetTscan DIA 3.0 acquisition. It also having DZ ion guide mass guard technology. This is and also the detector is photo multiplier ion detector. So this will give you more stability in the data, and also the life of the detector is much higher compared to the earlier model. So this is one of the 8600 is. Now I'll talk about the 8600 system because it is a new technology, and all the different workflow like DIA, DDA, PRM or MRM HR. This acquisition mode, it is possible to acquire the data using all this acquisition mode. Apart from that, we also have EA defragmentation application, not only restricted to proteomics, even for metabolomics, where you need higher level of electron kinetic energy for fragmentation. That is also possible using 8600, because from 7600, that is 0 to 25 electron volt tunable kinetic energy option was there, but here in 8600 it extended to 50 electron volts. So especially for lipids, that is very much useful. EAD, this tunable electron kinetic energy option in 8600. And then we have sensitivity. If you talk about sub-low attomolar level MSMS sensitivity, that is achievable through this 8600 system. So not only MSMS, even MS sensitivity also improves significantly if I talk about the lower model like 7600. And then quant accuracy and reproducibility, it is this system can offer around 5 orders of linear dynamic range, and then CB percentage, especially for DIA kind of analysis where CB, for differential proteomics analysis, CB percentage plays important role. So here you can say that very low CB, like less than 5% in many cases, CB percentage you can able to achieve across different level of proteomics analysis, like low abundant, high abundant, or middle abundant proteins. CB percentage is very good. Uh, we have different workflow called ZenoSWAT DIA or ZT-scan DIA. So these are the data independent acquisition workflow. This depth versus quantifiable ID. So one thing is that if you get good quantifiable identification, but your identification number actually get reduced. So that is one of the challenge. But in this kind of instrument, you will get good depth of analysis, like very low level detection. Plus, those detected protein also can be used for quantification. And EAD capability. So this is one of the good part. If I compare with the ETD technique, electron transfer dissociation, here you will get some additional advantage over ETD. One is the tunability. So from smaller molecular weight like metabolite lipids, as well as for glycopeptide, disulfide bonded peptides, or routine peptide analysis, the EAD, the electron kinetic energy. So you see electron-based fragmentation technique, where the electron kinetic energy can be varied based on the application. And it's a reagent-free process, unlike ETD. It is fast, so you can go up to 20 hertz speed in the DDA or DIA run. And then you can also couple CID and EAD combination called EACID. So mostly many application, as for example, glycopeptide, this is very much important. We got very good coverage sequence, MSMS fragment ion coverage of this EAD CID combination. So this is one of the good part in 8600. It is there in 7600, but 8600, the sensitivity improvement definitely will give better quality data. Then we have front end part that define nano LC, micro, low flow micro LC, all options we can able to couple. One is the EvoSafe, that is very high throughput nano micro LC system. Then we have water same class, that is also can be coupled with the ZenoTOF 8600. Then we have HDX system, that is also can be coupled with the 8600. And this source, that is OptiFlow source, this source actually can be switched from metabolomics or lipidomics. We can able to switch this. The capillary only have to change, so no need to switch the source for different application. So if I see the front end part, it is a DZ ion guide, which actually focuses the ions for better sensitive detection. And also we have IQ1 lens. This is a new things introduced in the 8600 system over 7600 or earlier models, where you can get better superior stability in the MS and MSMS data. And then we have a Q1 mass filter followed by EAD cell. So EAD cell is called electron activated dissociation cell. And then we have a CID cell. And then we have the time of flight analyzer. It's an N-shaped time of flight analyzer. Then this is the detector part. So this is all about the 8600 system. So you will get both CID and EAD fragmentation and xenotrapping. This is just after the collision cell, the ion trap is introduced in after the collision cell. So ion trap will improve the sensitivity in the MSMS level. And there are different software solution we have, like we have collaborated with different third-party vendors like Skylines, then Peak Studio, then and for this is for proteomics and MS Prager. Then for metabolomics, MS Dial, MZ Mine. These are the different software solution. Good part here is we can load the raw file directly to the software. In this software, we need not to convert the raw file into MZML or MZXML, different file format, which is compatible with this software. So it is quite possible to load the raw file from the generated from the system. So no need to go for data conversion. Another point is, once we are talking about very high speed data acquisition, so the speed of the instrument can go up to greater than 1300 hertz. So that is a very quite amazing MSMS acquisition speed. So if you run this high acquisition speed, the data file size will be relatively high. So to handle this bigger size of data file, we need a very good computing system. So here we have this high configuration PC we are offering for when we are offering this 8600 system. So these are the different features for 8600. The detector part, it is a optical detector, and this actually stability is pretty good, and optical with a 25 picosecond detection rate with a high speed pulse counting. So this is possible with the detector. So you can able to run this instrument with a very high speed data acquisition without compromising the data quality. Now coming to the different acquisition method, one is the DIA. So different DIA techniques, we already, like many people know about it. One is the DIA technique that use sequential MS acquisition. That means like SWAT or DDIA or DIA passive. So these are all sequential MSMS acquisition. But problem with this kind of acquisition, it suffer from overheads when switching from one mass range to the next mass range. So this overhead is needed to ensure that the ions transmitted in the previous Q1 mass range have cleared the Q2 collision cell. So this overhead occurs every time a mass range is transmitted. Therefore, the more selectivity or mass ranges that are applied to the DIA acquisition, the more this overhead cost. That means there will be a lot of this extra time required to perform this the sequential MSMS acquisition. So for high throughput analysis, this is a challenge. But then to address this problem, we have introduced in a scan called ZT scan. So this ZT scan is to switch from the sequential MSMS acquisition to a continuous MSMS collection using a Q1 scanning. So the quadruple one, it will be scanning continuously, and this is called scanning swath or diagonal passive. As MS data is being constantly acquired, the overhead from the clearance of the collision cell is not required. So this is not only speeds up the whole acquisition type, providing faster cycle time with more MSMS events for deeper sample coverage, but all the fragment ion retain the Q1 dimension information, enabling us to accurately associate the product ion with their respective precursor. So this is one of the better parts, the Q1 ZT scan here. The Q1 is continuously scanning, and can be the parent ion information can be connected with MSMS spectra. So this is very much useful for high throughput analysis, and it offers good sensitivity also, especially when we are talking about spatial proteomics or single cell proteomics. This kind of ZT scan workflow where quadrupole is also scanning, parallel TOF is also scanning. So parallel things are happening. So, so these are the different, like if you talk about the ZT scan, is very useful for low abundance sample, and it can be used for quantitation because it offers better quantitation when matrix interferences will be very high, and it offers very cleaner spectrum and higher throughput analysis. And for biological, like complex biological sample, this ZT scan 3.0, that is the new launch in 2026 ASMS, that is with 8600 system and also applicable for 7600 plus system, which actually reduces the window size from close to 1 Dalton window. That means almost nearing to the triple quadruple selectivity and specificity. So precursor fragment specificity via scanning quadruple dimension. So total speed can go in 8600, can go greater than 1200 and greater than 1200 hertz. So Q1 selectivity down to 1 Dalton with Q1 link precursor to MSMS. So this is the one of the good part in ZT scan DIA. Now coming to some uh, workflow for single cell analysis. So single cell using mass spectrometry. So single cell sample preparation plays important role. So there are many steps involved for single cell analysis, because single cell is actually the quantity of the protein. If you are talking about single cell proteomics, the quantity of the protein will be in very picogram level, like 100 to 125, 150 picogram level. So the first point is from a cell line, if you want to, or from a tissue, if you want to see isolate the single cell, that is the first point we need to do, like single cell isolation. Second is the uh, visualization of the single cell. So using the bright field and or fluorescence, we need to see where is the actually cell is located. Then another is the precision liquid dispensing. That is also very much important, because you need to dissolve the sample in a very high automated or high throughput manner. Then image recording of every isolated cell. Then temperature control also required for this kind of analysis. And end-to-end sample preparation workflow is mandatory. Now I will talk about some workflow here. Selenium, that they have this Proteochip Evo 96. So this is a sample preparation, because I am stressing on the sample preparation is very important for single cell analysis. So here you can see that there is a this workflow depicts this how we can able to transfer the single cell protein content from the tube to the LCMS. So the first step will be the collection of the bulk cell suspension, and then filling loading in the 96 well plate format is called Proteochip Evo 96. And then we need to isolate the single cell by visualization, by looking by microscopic visualization, and then picking the single cell from a mixture of many cells. And then we need to add lysis and digestion buffer mix. There's a mix of lysis buffer as well as digestion buffer that need to be added to the single cell. And then digestion, trypsin digestion or proteolytic digestion will happen. And then after that, dilution, and then this need to be transferred to the evo tube. So evo tube is a special desalting or sample concentrating tips that is used for sample purification. So once this dilution happen after digestion, that will need to be transferred to the evo tip, which actually desalt and then transfer into the LCMS column, and MSMS acquisition will start from there. So these are the different steps involved for single cell analysis. Next is what kind of uh, plate is important for this kind handling this kind of very small amount of sample. So this is one example where with up to 190 cells per run can be prepared in 3 hours time with the evo 96, with a high throughput proteomics with minimal hands-on time. So it's a completely high throughput manner we can able to process the sample. And then centrifugation based transfer is also important to evo tips, eliminates unnecessary handling. So we can do the centrifugation way. The process of centrifugation will help to transfer the sample from the to the evo tip. So that is one of the important. So this actually reduces the loss of the sample during the sample handling, and preserve the signal for deeper proteome coverage. This is one of the exercise done in Matthias Mann lab in ETH, the Matthias Mann in Max Planck University. There, what they shown that this is the like HeLa cell. and this is collected in PBS, phosphate buffer saline, with a 200 cells per microliter, and then it is loaded in 384 well plate with a preloaded 400 nanoliter of lysis buffer. And then they use Mantis liquid dispenser that used for transferring the sample in the 384 well plate format. And these are the buffer composition they use for, this is the trypsin as well as the lysis buffer is a mix, and that is used for loading the sample. Now, if you see the workflow here, in here, after that, this cell dispensing, and then we this is heated around 50 degrees for 90 minutes, and then digestion was performed, and then it was stopped by adding TFA, trifluoroacetic acid, and then peptides were loaded on EVO tips. EVO tip is a EVO-safe LC system is a part of that EVO tip is there. There, it can be loaded directly and transferred EVO tip, and then EVO tip is used for sample concentrating the sample, and then that can be loaded directly to the LC system, nano LC system. So this is the different steps you can see here. Like single cell sample preparation can be done in 2.5 hours, and then directly transferred and cleaned up within a 10 minutes time, and then transfer into the EVOSAFE LC system here. So these are the different things like pipette tips, EVO tips, different solvent, all these things you need to arrange before starting the single cell analysis. EVO tip, one of the good part is it is used for sample purification like desalting or any kind of other like any detergent or anything can be purified using this EVO tip, and EVO tip can be stored for long period of time. So one single cell information will be there in one tip, and that can be stored for many days. So this is one experiment was done for 1, 3, and 9 days, and then the performance recovery is very good even with the 9 days of time. So we can store this tip. Once the sample preparation is over, we can store it. You can see that this is a simple centrifuge where this EVO tip can be used. We can use the centrifugation to purify the sample. So one at a time, around 96 samples can be purified through centrifugal force. And this EVO tip box can be kept in the LC system, on the top of the LC system, and then it will take automatically, the robotic arm in the EVOSAFE will automatically take the tip, and then the sample will be sitting on the tip, and the liquid from the EVOSAFE LC system will enter the tip and then take the sample out of the tip and will be injected to the LC column, nano LC column. So this is very user friendly and high throughput manner we can able to use it. Now, if I talk about the traditional nano LC system for single cell analysis, it may not be very good because single cell analysis demand very high throughput analysis, and so nano LC, the typical nano LC what is used in the market, mainly having the problem of slow cycle time, like say, or slow gradient delay volumes, and lengthy sample pickup time, and prolonged column equilibration requirement. Then sample loss is another problem, like surface absorption, then dead volume, peak broadening, and evaporation in the autosampler area, and inadequate trapping in the trap column or the desalting column, the online desalting column. So system contamination like carryover, this is also another issue with the traditional nano LC. EVOSAFE system actually have some advantage over the traditional nano LC system. It also utilizes the EVO tip, that is a disposable microcolumn, and EVOSAFE integrates this offline sample purification and peptide loading, and this prevents significant protein loss. This usually occur in traditional nano LC system, and the gradient also like 100 sample per day or 500 up to 500 sample per day. The template or the preset methods that is already there in the EVOSAFE system. So you can able to analyze single cell and large scale cohort studies. So because single cell also have like different cell having heterogeneity. So we need to analyze thousands of single cell in a large scale cohort studies. And also it is very robust because radiant formation happen at very low pressure. So mechanical wear and tear are very less here. And high mass spectrometry utilization, that means your LC system is very fast. So mass spec is not waiting for the LC separation to be the auto sampler part and other sample injection part, uh, that takes very less time. So mass spec waiting time for LC conditioning or LC injection, sample injection is very less. So that is why it is very fast. So some data I will quickly show you. The one is that done in same Max Planck Institute. They and try to analyze using 8600 system with this EvoSAPS LC system with the same sample preparation workflow they have used, and with a single HeLa cell to 3 and 10 cell combination they tried, and then you can see the data with 8600 around 4000 protein groups starting from 3000 to 4700 range. Average 4000 protein groups identified using this 8600 system, and then 3 and 10 cell pool also identified 5100 to 6200 protein groups. So there, and with a very this is a 40 SPD whisper zoom 40. So that means 40 SPD per that's 40 samples per day with a ZT scan DIA approach. This is ZT scan DIA 2.0 used. Even to 3.0 we have a data which can give much better result over this particular data set, what the result they showed. And with the ion optics column of 75 micron ID with 15 centimeter length column is used in this case. EvoSAP Inno is the LC front end system used in this analysis. So this is 8600 this analysis. Now I'm talking about the single cell proteomics type of like analysis, like load amount of K562 cell line that we use with ZT-scan DIA 2.0. So here we can see that with a very low load of 62.5 picogram, we can able to identify almost 4,000 plus proteins. Whereas in 125 picogram, which is similar to almost single cell level, we could identify more than 4,500 protein. And here you can see that 62.5 picogram, the percentage CV less than 20%, such kind of protein identified is around 3,400. So that means this protein we can able to use for differential proteomics analysis. So this kind of sub nanogram or picogram level analysis can be done using 8600 ZenoTOF 8600 system. So I am stopping here. And I am handing over now to the next speaker, Dr. Tushar Maiti from RCB. Yeah.
Yeah. So thank you, Dipankar, for basically setting up the platform for the next basically presentation. So I'll just share my slide. Can you see my slides? Anyone can answer.
Yeah.
Yes, sir, it's visible.
Yeah. Yeah. So good evening, everyone. So I think previous speaker, Dr. Dipankar, already described the utility of like the ZenoTOF system. So I will basically present some of the application what we have carried out last like a few years, particularly its usage in the biomarker discovery. So my lab largely work on in the different aspect of biology, or you can say, yeah. So different aspect of biology. So one of the important aspect is basically related to proteostasis mechanism of Parkinson disease, where we study the molecular mechanism, how Parkinson disease basically proceed or the progression of the disease using like the animal model as well as clinical samples. So another aspect of my lab is basically looking at the biomarker discovery of pregnancy related complications. So the pregnancy is basically the important steps in like human life, or you can say every organism's life. So here we actually study how the pregnancy during the process of journey of like the pregnancy, there are several complications which contribute to the adverse fetal outcome or adverse basically pregnancy outcome. So one of the component is called preeclampsia, another is preterm labor, another is gestational diabetes, or like you can say interuterine growth restrictions. So the lab is basically trying to understand the molecular signature associated with different adverse pregnancy outcome. So why you will be studying the pregnancy complications or associated with the preterm birth? Because if you see like the sustainable development goal, like which the target for 2030, so the idea was basically preventing the death of newborn under 5 children, reduce the mortality like the rate to 12 death per 100 live birth or fewer. So because like the early 90s, 90s, 90, if we consider approximately 50 death per 1,000 lives birth was observed. And like over the period till 2019, we could see that like it reduced, but like it is not reduced significantly. So approximately 22 per 1,000 birth are basically observed. So India actually contribute almost 20% of global neonatal deaths, and highest number in 2023. If you see that, like almost like now 4 million deaths are basically observed in our country. So 3.6 million among the 27 million babies born annually are in preterm birth. So average annual reduction rate is 0.1% from 2010 to 2020, which is astonishingly very, very low. And 25% of overall global global preterm birth related complication or death is from India because of our huge population. So associated with, and the preterm birth is associated with diverse multimorbidity patterns from age group from 10 to 18 years of age of a children. So if we see epidemiology of preterm birth, so like the Bangladesh is basically reported highest number of preterm birth, like 16.2%. Next, subsequently India, 13%. And then even though the advanced country, particularly in US or even in China, we could see that significant number of the preterm birth percentage. So 10%, and China is basically reported a very low percentage. But however, most of the countries basically the preterm birth rate is above 10%. So these preterm baths is basically contributed different scales. Like one is called extremely preterm bath, which is less than 28 weeks of POG, the period of gestation, and very preterm bath which is ranging from 28 to 32, and moderate after 32 weeks of pregnancy till the delivery 37 weeks is considered as late or the moderate preterm, and anything weeks we consider as a term birth. So if we see the concern, why we concern about the preterm birth? Because the complication associated with preterm births are diverse. So the baby suffers from pulmonary complications, so CNS complications, retinopathy, prematurity. Apart from that, the preterm baby is also basically display several types of the disease, including sepsis, congenital abnormalities, the respiratory problems, like again the diabetes associated complications, all of them basically observed in case of like the preterm birth babies. So that's why it is very important like to study or like important to prevent, even to find out something molecular signature which could actually prevent the preterm birth. So so biomarker for preterm birth, like because the as I mentioned, this problem is basically addressed like several years. It is not basically just initiated. Like almost like 40, 35 to 40 years, people are extensively working in the areas of like the biomarker discovery associated with preterm birth. So if we see the different types of molecular signature people have identified, like one is basically called salivary estrol, and again like there are many protein molecules like insulin binding protein 4 and sex hormone binding globulin. Their ratio is basically considered one of the important marker like which reported earlier in 2016, and this particular ratio has been like basically validated in several countries. However, the discovery of all these molecules are basically lacking because of their large number, because of their sample size, as well as basically technological disadvantage or technological problems. So low sensitivity and specificity of potential marker, inadequate sample size, and like many of them like basically showed very good promise in case of discovery studies. However, they failed in the validation studies. So another important aspect is phenotypic heterogeneity. So preterm birth is basically associated with many other complications, as I mentioned, like the gestational diabetes or pre-eclampsia or intrauterine growth restriction. So all these complications are basically like the mixed each other. There is no clear-cut segregated phenotypes for each basically condition. So that's why finding out a single molecular marker for this kind of conditions will be really, really a challenge. So, and most of the identified markers represent the symptomatic pregnancy. And the identified proteins, if you can see here, is largely based on the high abundant proteins. So because these proteins are basically detected earlier in multi mass spectrometry or the earlier basically mass spectrometry workflow, which could largely features or could identify or quantify the high abundant proteins. So next, as I mentioned, the preterm birth is basically classified one way, like the extremely preterm, like the very preterm, moderate preterm, and like the term birth. Similarly, preterm birth also classified based on the phenotypes. One is called spontaneous preterm birth, which is basically 60 to 70% of preterm birth is represented as a spontaneous preterm birth. That means there is no information or no, like now you can say, like the cause for this particular conditions are reported, and then like the subsequent the mother basically deliver the baby. So this kind of the preterm birth is largely reported as a spontaneous preterm birth. So among these spontaneous preterm birth, like now the 40 to 45% are reported as a idiopathic preterm birth, and like 20 to 30% are reported as a premature rupture of like the membrane. So the other category is called medically indicated preterm birth, which largely related to the conditions for preeclampsia, like obstetrical complication, placental abrupture, fetal distress, intrauterine growth restriction. So these are the conditions which shows some kind of phenotypic, like the display the phenotypic changes during the progression of the pregnancy. So one could actually get some kind of clinical correlates which doctors basically described as a preeclampsia or like IUGR or like the fetal distress or placental abruption conditions. So this is very I think this particular part like where you have some sign of informations there, like doctor can actually treat this kind of patient. However, spontaneous preterm birth, which is very difficult to treat, until unless something molecular signature or molecular markers reported early, and like early stage one could actually identify them and classify them as a spontaneous, the mothers can deliver in the spontaneous preterm birth. So next, the the study design of our basically cohort. So this particular study is embedded in our like a clinical cohorts, which is called Garvini cohorts. So it is basically a interdisciplinary cohort which was set up in 2014 by Department of Biotechnology, and the cohort was basically managed by Translational Health Science Research Institute at Gurgaon General Hospital, and many other clinical hospitals, particularly All India Institute of Medical Science, Maulana Azad Medical College, Safdarjung hospitals. These are the basically clinical sites also involved in this particular study. However, the patient recruitment largely happened in the Gurgaon General Hospital. So we established this cohort in 2015, and subsequently the sample collection process continued, and still the some basically study the samples are basically collected in collection is still continuing. So in this particular cohort, the we have basically so far collected approximately 12,000 basically mothers samples, and the different type of biospecimens like the saliva, the placenta, the blood, then again cord bloods, and then again large number of clinical parameters, ultrasounds, the images. So these are all basically collected serially in in these cohorts. So we have basically taken the samples based on the exclusion inclusion criteria, which is basically mentioned here. The exclusion criteria is comorbidity, PIH, preeclampsia, and HELLP syndrome, fetal abnormalities, HIV, the anti-retroviral therapy, malaria, multiple birth, stillbirth. So these kind of conditions are basically excluded from excluded from these the patient. And inclusion criteria is basically live single tone spontaneous preterm birth, and plasma samples should be available V1, V2, and V3. So it is basically we time point the visit 1, visit 2, and visit 3, which is representing at visit 1 less than 14 week. Visit 2 is representing 18 to 20 weeks, and V3 represent 26 to 28 weeks of pregnancy. So a longitudinal basically sample should be available. So in this case, we have actually taken V2 and V3 time points. The important is basically this V2 or visit 2, the 18 to 20 is basically the mothers undergoes large number of clinical tests. So largely the anomaly test and all other basically tests carried out in the VT time point. So that's why this time point is very critical. And if we can predict something in this particular time point, it will be very much useful for the clinician to find out basically the solution, or find out some kind of a strategy to stratify the mothers. So, and the 26 to 28 weeks is basically used for the patient where mother undergoes the OGTT test, particularly for the gestational diabetes assessment. So we have taken from the pregnancy our the Garvini cohorts. So less than POG less than 35 is considered as a spontaneous preterm birth, even though less than 37 is considered as a preterm birth. But in discovery cohort, we have actually taken less than 37 because we want basically clean the population where we can actually confirm that the mother which is basically undergoing delivery in our cohort is basically representing as the proper a spontaneous preterm birth. So anything less than greater than 80 say 39 is considered as a term birth in our basically sample analysis. So we have taken 27 pair for the every time points the preterm versus the term birth, and then subsequently we perform the proteomics experiment. So the workflow basically Dipankar already mentioned that the data independent acquisitions basically workflow. So the sample weight preparation is as already Dipankar mentioned that it's very critical step. So we have taken the plasma with dilutions we actually used approximately the 50 the microgram of basically protein, and then trypsin digested, and then subsequently clean that samples using C18 cartridge, and then we injected the samples in the the micro LC micro LC setup with ZenoTOF 7600 systems with SWAT the DIA workflow. And once the data is basically acquired, the data was analyzed in the Spectronaut which is basically the software available from the third party, and then data was analyzed. And these like the same times this data is also like analyzed in the spectra note, and we also took the data, and then subsequently the identified the data is basically taken, and then subsequently we perform different type of feature based selection algorithm, and then subsequently correlate with the differential proteome against the feature based selection method, and from that we actually selected the hit like the hit proteins or the our target of interest. And this target is basically validated in the multiple reactions monitoring the mass spectrometry using like high resolution like mass spec, again the HRMRM system in ZenoTOF mass spectrometer. And subsequently we have actually model build and then classified whether this particular the molecule can predict the preterm birth with the term birth with sufficient accuracy and precision or not. So using the separate basically subset of samples in the validation cohorts. So if we see like here, the total proteins we identified like the approximately 630, and around 800 nanogram protein is basically injected in the micro LC setup. So we identified 630 proteins with 85% completeness with 21-minute gradient. So in the table, like this is the data published by Journal American Society of Mass Spectrometry 2024, where they have given the comparative analysis of different mass spectrometric instrument, where you could see the different gradient setup as well as the data analysis platform and the number of proteins basically identified in the system. So if we could find Q executive system from Thermo, Team Top system from Bruker's, 66 like the triple top system from like the with water M class system from Sykes, and like that again the Team Tops Pro systems with different gradient from 90 minutes, 60 minutes to 11.5 minutes gradient basically used. And what you could actually find out that the number of proteins so far in this particular paper reported 664 using the Q executive system with basically 60-minute gradients. So with approximately, if you can say that 100% completeness of the system is only 241. However, in case of the Q executive with 90-minute gradient, you could see that 484 with the 50% like the completeness. So like if you can consider the completeness of 100% completeness, if you consider that like the even basically Team Tops Pro, it could actually give like very low the coverage basically using basically shorter gradient. So in this case, like we identified approximately 630 protein with 85% completeness, and which is basically really good number using these GenoTOF like systems. So when we have compared between the V2 and V3 time points, then we could actually see that approximately 552 proteins are common between both basically time point conditions, and we consider the fold change 1.5 fold as a upregulated and less than 0.66 as a consider as a downregulated. And like I have not basically put this information there. The data quality has been already verified, checked, and most of the cases the data completeness is basically, as I mentioned, 85%, and the CV percentage is less than basically 30%. So as it is a clinical samples, basically the inter-person variability will be always there. So clinical samples less than 30%, like now, like we believe that it is a very good basically consideration, very good basically sample quality. So V2 and V3, if we consider, the upregulation is 90 protein and downregulation is 20 protein. Similarly, V3, 80% is upregulated, 80 proteins are upregulated and 10 proteins are downregulated. So then next what we have done, we have taken the 630 protein, which is basically identified and quantifiable peptides. So their intensity has been taken, and then we put the Python algorithm that like use the machine learning algorithm with different types of basically the learning packages. Here it is mentioned the SVM like LR or KNN. So different packages are basically there. So then this basically the molecule, this particular particular samples and the identified proteins, we actually validate, like stratified in the five crepe-fold cross validation, and and data stratification is basically carried out. So here you have train set and basically testing sets. So, and this particular the cross validation strategy basically provide basically the specific signature which is associated with the which can actually segregate term versus p term. And then again, this model is basically validated in different classification model like SBM model, logistic regression, random forest, and feature extraction algorithm was used, recursor, feature elimination, and SARP. So these two basically feature extraction method has been used, and then subsequently basically the feature have been placed for V2 and V3. So like if we see here that the two feature selection extraction methods have been taken, and then what we did, we mapped these the differential proteomes with the extracted features. So whatever extracted feature we have 53 plus 42, these features are basically extracted features, and we have the differential proteome. So if we combine them, like we have a common 42 features, which is basically both identified in the differential proteome as well as feature extraction method. Similarly, V3, we could get 29 features, which is basically present both extracted features and the differential proteome. So the features are already basically described in this case, and where both basically feature extraction and differential proteomes are displayed. And the star basically represents the star represents like in the every bar represent these proteins are basically present in both V2 and V3 conditions. So next, what we have done it we actually took that particular proteins basically different type of transitions, and then subsequently we verified with this our samples the identified basically features is basically subsequently perform the multiple reaction monitoring in HR high resolution mode, which is HR MRM mode, and then we actually try to find out that which particular the precursor ion or basically transition is giving some kind of basically classification which describe the p term versus p term. So the feature extraction method here we use the Boruta and the training sets, and then we could actually get the two important features which is giving the caps the caps 2 protein and Michael 2. These two proteins is basically showing a distinct features which is basically present in the verification state. So next, what we have actually done, we have taken the 733 the non-case sample and 52 the case samples. It is basically case cohort design. So case the earlier it was basically case control design. So now we have taken the case cohort design where the 700 the 32 these particular proteins represent the non-case. The non-case means it carries all other kinds of basically the comorbidities that are associated with it, and 52 basically the samples are representing the spontaneous p number. So now if we actually this represent the case post cohort design represent more or less the realistic features what generally we see in the clinic. So sample preparation is carried, data processing, then subsequently we actually took the V2 time point, 18 to 20 weeks time point, and then the features basically which identified earlier, then we perform the HR MRM in the ZenoTOF mode, and then this basically data was analyzed in the peak integrations, and the peak normalizations were carried out in the multi-point. So next, basically the features what we have actually again taken. So here the red color represent the right hand side representing the classifier which could actually described by these the Boruta algorithm, and these molecules or these particular features could be properly segregating the p term versus term birth transitions. So next, we actually try to perform the predictive models using this particular, the red color, the right hand side, whatever the transitions you have mentioned, largely the proteins as I mentioned that CAPS2 and the CAPS2 proteins as well as Michael-2 proteins. So using the different transitions, like we actually try to build the models. So performance model was basically carried out CAPS2 and Michael-2. So both these two proteins are basically combined and try to find out that specificity and negative predictivity and the sensitivity is basically plotted, and different machine learning algorithm was basically used. So what we could actually find that the negative predictivity is for SBA model, logistic regression, random forest, all cases we could see around the 94%. However, the positive predictivity is basically 7.6%, 5.9, 7.1, and 6.7%. However, the sensitivity of this particular test is drastically low, 55%, even 13%, 51%, and 48%. So the best performing model is basically SBM, which actually display that positive predictivity 7.6%, 94%, sensitivity is basically 55, and specificity is 52. So we expect here that high sensitivity and specificity. That means if our sensitivity is approaching towards basically 100 and specificity is also towards 100. This is the ideal conditions, which could actually, if we achieve this particular condition, then we can say that this will be a positive predictivity. And second thing, as the low positive predictivity, one could actually say that why we are getting 7.6%, because the cohort, as we mentioned, it is a combination of basically case and non-case. So our p-term birth rate is basically approximately, as earlier I described, the 13%. So we are very much close to the positive predictivity. That means 13, approximately the abundance, the p-term birth abundance, we could actually reach this kind of percentage. However, the sensitivity of this particular test is basically very low. So which negative predictivity also, we cannot use, even though negative predictivity is 94%, but they have significant number of the false, basically the negatives will be there, which cannot be basically predicted in this particular model. So next, subsequently we have carried out we try to find out that what is the importance of this particular molecule called CAPS2. So recently people have actually demonstrated genetic mapping susceptibility of spontaneous preterm birth analysis in UTA pedigree analysis and inherited genetic factors. They described here that the CAPS2 basically something basically the polymorphisms basically linked to the preterm birth. So that gave some certain kind of hints that yes, this molecule could be ideal molecules, can be used for the subsequent steps. So next, we actually thought of doing the predictive models two, where we use the CAPS2. So if we use the CAPS2, the SBA model, if we actually see the positive predictivity didn't actually gone down or increase. However, negative predictivity percentage is basically high. The one interesting thing is that sensitivity is significantly increased to 65%, indicating that this particular cases our basically model performing the negative predictivity is performing certain basically extent better if we only use the CAPS2 molecule. So next, we actually we have checked the distribution of our sample. If we could see that 37 is basically the considered as a term birth. So majority of our sample is basically associated with term birth. Very less number of samples are basically associated with the preterm birth. So, or with extremely preterm birth. If we stratify the sample like the extreme preterm or moderate preterm and term birth, then how this particular model will perform. So then, we actually again predict the model. So model 2 with high risk spontaneous preterm birth. That means the patient which is basically showing less than 35. If we use them, then what the SBM model demonstrated that positive predictivity reduced little bit. However, the negative predictivity is basically enhanced 97%, and sensitivity is basically significantly increased 77.41%. So this indicate that these particular markers basically can be used as a specific screening marker. and used for basically preterm birth the screening. So how this particular molecule can be used as a the screening marker? So now I'll just give you here the information. So if you have 100 patient screening in the second trimester training, so in that case could be 900, the 60 patient, if we consider the high risk pregnancy is 40%, 4% only, so 40 patients will be undergoing the spontaneous preterm, and 940 is basically the 960 is basically considered is the term mother. So now if you have a 90% negative predictivity, that means in these conditions you have a false negative of 4 and false positive of a, and the positive of basically 36. So these 36 basically can be referred to the other the testing centers or other clinical parameters can be checked and basically will be monitored. And this false negative is basically 4 can be basically incorporated in this particular study. So similarly 690, if it is basically the 40%, now if we can say the specificity is 40%, then the false positive will be 576. So this 576 mother, which basically shows the specificity of 40%, this can be transferred to the next basically platform where the patient will be basically managed and further test will be carried out. So at least in this particular population, among the 1,000 patients, you will be able to basically around 600 patients can be basically further the screened and then basically referred to the next level. You can completely eliminate 400 patients based on these basically negative predictive value. These 384 patients which basically negative predictivity is 99%, which is completely free of basically the preterm birth, and that the patient does not require any kind of referral. So at least you will be able to reduce the substantial burden from the preterm birth screening. So our study basically here, we actually proposed that the CAPS2 molecule can be used as a screening marker and can be used for basically the further basically screening and can be used for the clinical settings. So this particular work is basically presently at that moment in bioarchive, and it is under review in a peer-reviewed journal. And now the one of the important basically aspect I want to mention here, this is basically the targeted image, but we have not basically performed the absolute quantification of protein. So presently we are basically collaborating with Max Planck Institute with Matthias Mann lab as well as basically other consortium where a large large number of discovery studies also coupled with our study, and we have basically selected approximately 50 the biomarkers which will be further quantified using isotope level proteins and used for the India centric validations in different cohorts, different cohorts in our country. So I will stop here. First of all, this is my lab. Largely this work is basically carried out by this person, is Naman Kharbanda, who is presently basically doing the industry job in the mass spec something service company. And thank you very much for your attention.